All right, this is a Chapter 6 review. This is the first video in the Chapter 6 review for Exam 2. Let's go over mitosis and meiosis and the similarities and the differences between them. Mitosis, the purpose of mitosis is to create diploid or diploid daughter cells. Remember that in a chromosome, or in a human cell, you've got a total of 46 chromosomes. So that total number of chromosomes in a normal cell is considered the diploid or the diploid number. And the diploid number is going to be different for each organism. We all kind of have a different number of chromosomes. And so it's different from organism to one organism to the next. But the total number of chromosomes that you have in a normal cell, a normal human cell, is considered the diploid number of chromosomes. And the reason it's diploid, because di typically means two, is because you've gotten half of those chromosomes from your mom and half of those chromosomes from your dad, and those chromosomes are complementary to each other. So the ones that look the same, where you get one from your mom and one from your dad, those are considered a homologous pair of chromosomes. And so you've got 23 pairs of homologous chromosomes in a human cell, so 23 pairs equals 46 total. So the diploid number is the total number of chromosomes. In meiosis, the purpose is to create haploid daughter cells. So cells that have half the number of chromosomes. And so basically you're going from a diploid or a 2N parent cell, and you're making 1N or just N daughter cells, haploid daughter cells. And you're starting with one parent cell, and you're ending up with 4 daughter cells. And that's what's going on in meiosis. And in that process where you're halving the number of chromosomes into the daughter cells, because of that, the daughter cells are going to be different from the parent cell because they have half the number of chromosomes. They're also going to be different from each other. And the reason that that occurs, we'll actually get into a little bit more when we discuss the process of meiosis, but basically there's some crazy things that go on amongst the chromosomes where they can exchange bits and pieces and they can be different, and it causes some genetic variation from one generation to the next. So the haploid daughter cells in meiosis are different from each other, and they're different from the parent cell. In contrast to that, in mitosis, we're starting with a 2N or a diploid parent cell, and we're ending up with a 2N or a diploid, two of them actually, diploid daughter cells. So you start with one parent cell and you end up with 2 daughter cells. And those daughter cells, because they are diploid, they are identical to the parent cell, and they are also identical to each other. They have the same number and complement of chromosomes to each other as to the parent cell. So the overall purpose of mitosis, if you're making identical cells, is really either growth. So a small organism, as it gets bigger, is going to undergo a lot of mitosis, so it's growing new cells, and also for healing. So if you injure yourself, your body makes new cells to replace the ones that were injured. And any cell that's made through mitosis is a body cell, so it's called a somatic cell. And basically that's everything other than your sex cells. So the somatic cells are every cell in your body other than your sex cell. So in a human, that would be the 46 chromosomes that would make up a normal somatic cell. So before mitosis, the parent cell has 46 chromosomes. After mitosis, the daughter cells still have 46 chromosomes. That's different from meiosis. Meiosis, the general purpose of meiosis is reproduction. So you're making sex cells, as in contrast to the somatic cell that you see in mitosis. So for humans, those sex cells will be the sperm and the egg cells. And these are termed gametes. In the human life cycle, the sperm and the egg come together, and when you combine those, you form a zygote. So in a human, the parent cell is going to have 46 chromosomes, and then the daughter cells after meiosis would have 23 chromosomes. So each sperm and each egg cell has 23 chromosomes. When they come back together to form a zygote, they fuse and those chromosomes come together, and you've got your full complement again, your diploid number, your 46 chromosomes. So that's the general purpose of mitosis versus meiosis. So make sure you understand that, because there will be some test questions over kind of how they're different and how they're similar. The next thing we're going to look at is we're going to go into the stages of mitosis. Mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. And then cytokinesis is the separation of the cells, the separation of the cytoplasm. And that's a little bit different. This picture kind of shows both of them happening at the same time, but that's not technically considered part of mitosis. Remember, mitosis, the point is to make identical daughter cells. And so as we go through this process, keep that in mind, that the parent cell shown here kind of after the DNA is replicated, the parent cell is going to be identical to the daughter cells down here at the end. Now, as I said, remember that the DNA has already duplicated before mitosis occurs. And so that the cell that you see here, the nucleus you see here, this is kind of double the amount of DNA that would be present in a normal adult cell for this particular organism. So this organism has 1, 2, 3, 4, 5, 6 chromosomes total, but each of those chromosomes has already duplicated, which is why it looks like an X. So each part of the X is a sister chromatid, and they're identical to each other. So the two, on this red X right here, the 2 parts of the X are basically the chromosome that's duplicated and is bound together at the centromere. So the sister chromatids are bound at the centromere. And remember, like I said, this has already happened. This duplication has already happened before mitosis starts. In this particular cell, which has 6 chromosomes total, they're color-coded. 3 of them are red and 3 of them are blue. And the purpose of that is that you can tell which ones came from the father and which ones came from the mother. So let's say the blue ones came from the dad and then the red ones came from the mom. Remember that, like in the human, you've got 46 chromosomes total, but you've got 23 pairs of homologous chromosomes. So in this particular cell, there would be 3 homologous pairs. Here's one pair, the red and the blue are the homologous pair. Here's another homologous pair, and then here is a 3rd homologous pair of chromosomes. The reason there are so many sister chromatids in these homologous pairs is because the duplication has already occurred. So because of that, this homologous pair, which you would think would just have 2 chromosomes, actually has 4 sister chromatids because of that duplication. So in prophase, the first thing that's going to happen is that the chromosomes are going to condense down. And what that means is that you can actually see them. When you're looking at a normal cell at the resting state, you cannot tell the chromosomes apart. It's just kind of this mishmash in the nucleus. So in prophase, those chromosomes actually condense down where you can see them if you're looking at them with a microscope. The nuclear envelope, which is the coating around the nucleus, breaks down, and that's going to allow the chromosomes to kind of move around in the cell. So the envelope is no longer there, and then structures called microtubules will form and grow. And those microtubules are going to end up forming the spindle, which is going to be at either side of the cell, the spindle apparatus, and it will be involved in kind of separating out the chromosomes when that time comes for mitosis. All right, so that's prophase. In metaphase, the chromosomes are going to align at the center of the cell. And this is called the metaphase plate, where they line up. And it's just the center of the cell. So think of metaphase and middle. They both start with M. So the chromosomes line up along the metaphase plate here. And notice that so like this red X at the top, it's lined up completely up and down. And so the sister chromatids are kind of to either side. And so what's going to happen in the next phase of mitosis and anaphase is that the spindle apparatus, these microtubules that are attached to the centromeres, are going to pull them apart. They're going to pull apart the sister chromatids. So in metaphase, the chromosomes are lined at the middle of the plate, and the centromeres, which are what join the sister chromatids together, the centromeres are attached to microtubules. And so down here in anaphase, those microtubules are going to shorten, and that's going to pull the sister chromatids apart. And this is kind of the shortest phase of mitosis. During this phase where the sister chromatids pull apart, you'll notice that as they go to opposite ends of the cell, that this cell still has 1, 2, 3, 4, 5, 6 chromosomes. And this one's going to have 1, 2, 3, 4, 5, 6 chromosomes. So the sister chromatids, which were the duplicated chromosomes, get pulled apart in anaphase. And then the last phase of mitosis is telophase, and this picture kind of shows a combination of telophase and cytokinesis. But in telophase, the nuclear envelope is going to reform around each new set of chromosomes, around each new nucleus. So the nuclear envelope is going to reform, and the chromosomes revert back to their uncondensed form. Now, cytokinesis is a little different in animal cells versus plant cells. This picture is showing an animal cell. So in an animal cell, in cytokinesis, a band of proteins will form around the center of the cell and kind of pinch the cells apart. And so you can see that in this picture, that there's this band of contractile proteins that's going to pinch these two cells apart. And each of the daughter cells is going to be identical in the number of chromosomes to the original parent cell. And just remember that in this picture, in prophase with this parent cell, the chromosomes had already duplicated. So before this duplication occurred, there were 6 chromosomes, and at the very end, there will be 6 chromosomes in each of the daughter cells. In plant cells, cytokinesis is a little bit different because remember, plant cells have a cell wall. And so in cytokinesis, what happens is a new cell wall is built between the daughter cells, and that's built on the metaphase plate, which we call the cell plate when it gets to that stage in the process. So in a plant cell, you would actually just kind of build up this wall between the cells on the cell plate or on the metaphase plate. So it is different. Cytokinesis is different in plant cells versus animal cells.